Today we will study the diagnosis and control techniques of plant viral diseases. 1. Viruses are a group of organisms that are encapsulated in a protective capsid of proteins and lipoproteins and can only replicate their genome in suitable host cells. Viruses that infect plants are called plant viruses. The main characteristics of plant viruses are 1. Very small non-cellular organisms, visible only by electron microscopy. 2. Simple structure, nucleic acid plus protein or lipoprotein capsid. 3. They are living parasites and can only survive and proliferate in living host cells. General traits of viruses. The morphology of plant viruses is granulomatous, with most viral granules being spherical, rod-shaped, thread-shaped, and a few being bullet-shaped and duplex-shaped. The size of virus granules is calculated in nanometers, so only electron microscopy can be observed. 2. Virus structure. A complete virus pellet is composed of nucleic acid and an outer envelope of protein. Plant viruses have two basic structures, rod-shaped and sphere-shaped respectively. The nucleic acid is in the middle and the protein is enclosed outside the nucleic acid, forming a coat that protects the nucleic acid. 3. Stability of plant viruses. The temperature at which the virulent sap maintains its ability to infest within 10 minutes is the minimum temperature at which the viral sap loses its pathogenicity. Most plant viruses have a passivation temperature of about 60 degrees Celsius. 4. The transmission of plant viruses can be divided into mesophilic and non-mesophilic transmission. 1. Non-mediated transmission. It refers to transmission without the intervention of other organisms such as with sap, pollen, grafting, and asexual propagation materials. Also known as sap friction transmission. Mainly mechanical contact resulting in microscopic injuries leading to virus invasion. Direct transmission of the virus by pollen is relatively rare, but can be carried by bees. 2. Vector transmission. The virus is transmitted and infested by other organisms such as insects, mites, nematodes, fungi, etc. through the activities of the organism when plants are infected with viruses. Most of them show the characteristics of viral diseases, which can be diagnosed based on the typical symptoms of viral diseases. In terms of symptoms, the main symptoms are leaf blossoms, yellowing, dwarfing, bunching, and necrotic spots. The symptoms appear first on the heart leaves and then extend to the whole plant. 6. Control methods of viruses. 1. Plant quarantine. In agricultural production, seeds or seedlings carrying viruses are the source of plant virus diseases. The transfer of seeds and seedlings from infected areas to other places is strictly prohibited. 2. Breeding non-toxic seedlings can be done through heat treatment and stem tip detoxification. 3. Selection and breeding of disease-resistant varieties. According to the main sources of local viruses, choose disease-resistant varieties according to local conditions. 4. Strengthen field management. Agricultural operations to reduce branch collisions, mid-tillage to reduce root injury, remove diseased seedlings, and disinfect with soapy water or 10% trisodium phosphate solution. 5. Crop rotation can be done in water and dry rotations. Physical control. Virus disease can be prevented and controlled, but not cured. Mostly transmitted by stinging mouth-breathing pests such as aphids and leafhoppers, etc. Advocating the use of insect-proof nets for seedlings and cultivation. Taking advantage of the yellowing nature of aphids and whiteflies, you can hang yellow sticky boards or cover with silvery gray mulch to repel aphids. 6. Pharmacological control to prevent the occurrence of virus disease. We need to prevent and control the stinging-sucking insect pests. If necessary, we can use imidacloprid, thiamethoxam, imidacloprid, imidacloprid furoxam, acetamiprid, etc. in the case of mild viral diseases. Mushroom polysaccharide, amino oligosaccharide, morpholinoguanidine hydrochloride, ninhydrin, toxaphene, morpholine copper acetate, etc. can be sprayed. Spray once every 10 days, 2 to 3 times. Students, this lesson is over. Thanks for watching.